Like I said, I really just want to thank you all for joining us today. We're really excited to have you here and talk about some of the things that are going on at PacBio, both inside the company and with some really fantastic researchers and clinicians outside the company. So what we're really going to talk about today is what we believe is a new era in rare disease research, everything from what we're offering on the product side, through the analysis, into the clinic and into population research. And so I'm going to kick it off. Like I said, I'll give you a brief introduction of some of the things we've announced recently at PacBio and you may have heard about here at the conference. I'll then throw it over to my colleague, Mike Eberle, our head of computational biology. He'll talk you through what you can do with the data, how to get the most richness of information out of it. And then we'll wrap up with two great talks from Carol Saunders at Kansas City Children's Mercy and Mait Metspalu from the University of Tartu, really showing how you can use that data and gain really actionable and exciting insights out of it. So with that, I'm going to jump in, and I thought I'd start with our mission statement, and many of you have probably seen this, but I really think it's important to start here because that's exactly why we're here today. We're here to enable the promise of genomics to better human health, and hopefully over the next hour or so, you'll really see what we're doing on the product side to enable that and how customers are really taking advantage of it and putting it into practice. So it's been a really busy year for us over at PacBio. When we were here at ASHG a year ago, we'd started speaking about our Connects kits. And since then, we've really launched a number of products, analysis options, data sets, and collaborations that really help you get the most out of this rich, exciting data type that we have called HiFi. And so even as early as Q1, we were launching new prep kits, including our Pure Target offering that allows you to look at repeat expansions. Mike will talk a little bit about what you can do with that. It allows you to look at those without amplification, so you get a really unbiased and exciting view of those specific targets. In Q2, we launched a number of other kits to look at full-length isoforms and RNA sequencing, as well as bringing out databases like ColorsDB that allow you to try and understand the frequency of some of the structural variants and other variant types that are cryptic to short-read technologies and are able to be seen in those dark regions of the genome that PacBio and other long-read technologies allow you to access. And then just last quarter in Q3, we shared some of the great work going on with the HiFi Solves Consortium. Again, you'll hear a little bit about this today, really demonstrating how this exciting data type is being put into practice and changing what happens with regard to rare disease research and getting answers for so many of those families. But what I'm going to spend about 5 minutes on now is just talking about everything we've announced over the last sort of 10 days here at ASHG, because I really think it is enabling and allows you guys to think about the science, the questions you're asking in a different and ideally more open and unbiased way. So coming into ASHG, we'd made announcements about 3 key areas of our workflow: the upfront extraction with our nanobind technology, which came out of the Circulomics acquisition we did a few years ago, our SmartLink cloud solution that I'll talk about that enables you to manage and process your data in a new way, and then finally talk about some of the advancements we've made on the Revio platform, which we actually launched at ASHG 2 years ago. So jumping straight in, earlier this year I mentioned we launched our pan-DNA kit, and the idea behind this kit, and it was the first kit that PacBio developed after the Circulomics acquisition, was to enable a single kit that really lets you tackle just about every sample type out there so that you could get the highest quality, the highest molecular weight DNA out of any of these sample types and take that through into HiFi sequencing on any of our platforms to get great results. Now, as you can imagine, being in a post-COVID area and all the things that came along, there's one sample type that was missing here, and that was really saliva. So we were really excited last week to launch our saliva protocol, which is now compatible with this kit that's on market. So if you're interested in sequencing saliva and getting long reads out of it, you can now use this kit and this protocol. And as part of that, we added Origin to our compatible partners program. So these are the kits we support with regard to collecting the saliva and extracting it. And what's been really exciting, especially for me who spent some time at the Garvan Institute, is seeing how people are taking advantage and using this and getting fantastic results out of it. So you can see Igor here talking about the saliva extraction, and this was from some older and really challenging samples that were collected in remote parts of Australia, whereby they were able to get out high molecular weight, high quality DNA, and go on and generate beautiful HiFi genomes from these really precious and exciting samples. So we're thrilled to see how well this is working, but when we started digging into the data, there's some really cool things that come out, and this is one that I love to point to. As you can imagine, there's a real proportion of the sample that you get when you collect saliva contains mouth flora or bacterial contamination. At the same time, one of the beautiful things about HiFi sequencing is that you get methylation performance and methylation calls out of that data free. And so if you combine these two things, what you can see in this little plot here is that at the end of the run, you'll get a measure of methylation from your genome. And that measure, as you see on the y-axis there, listed between 30 and 70%, really beautifully correlates with the percentage of the bacterial fraction in your sample. So right at the end of the run, if you see that you've got about 60% methylation, you know you're probably at about 5% bacterial contamination in there. If you're down at sort of 30, 40% methylation, you're probably in the 25, 30% bacterial contamination. And so not only is this a great QC, so you know what to do with the sample next, whether you want to go recollect, resequence, go deeper, or just accept it, but it's the fact that it preserves the biology so beautifully coming through that's really exciting. So this was really encouraging to see. But the other thing we found that was really, really exciting about these collection kits is that when you collect them into this buffer with the origin kits, the DNA becomes incredibly stable. And so if you look here about, over a year ago now, back in July of last year, we collected a sample inside PacBio, and we asked our wonderful R&D team to just patiently leave it alone. Please don't touch it. We need to age this and see how it performs. And so they graciously agreed not to do some work for a change. And a year later, so 352 days later, we extracted this with this protocol, ran it on Revio, and were able to get 120 gigabases out of that sample. So not only is it great for making access to these samples and collection of these samples easier, it also allows us to go back to samples that have been stored or collected earlier and get fantastic results out. So that's the first exciting thing that we announced coming into ASHG, and I was keen to share with you folks. The second part is what we've been doing on the SmartLink front. So for those that aren't aware, SmartLink is our web-based end-to-end workflow manager. It's really what allows you to manage the sequences, monitor the runs, look at how you're setting up samples, manage your data, et cetera. And it's really a key part of our workflow and a key part that allows people to scale up and get running with PacBio sequencing. And historically, we've always offered this in two flavors. You've had the SmartLink Lite, which is sort of a desktop solution. It doesn't have the analysis features, but allows you to do all that run management, but you still need a compute server for it. Or we've had the fully capable SmartLink solution, which is usually installed in a HPC cluster, and that includes things like analysis. But coming into this week, we announced that we're moving all of this to the cloud. And so for the first time on PacBio, we now have SmartLink in the cloud. This is something that you'll be able to sign up to just using a regular Okta-type authentication service, create work groups so you can manage and share your data. But this is all cloud-enabled now, which is really exciting for us. A couple of things that this allows us to do is to have a multi-cloud compatible workflow. So what this means is you sign up, you can do all your run management, et cetera, but you get the choice of pointing the instrument at any of the clouds you want to use, whether it's Google, Amazon, Azure, et cetera, whichever one you're using in your institutes and facilities, and take advantage of all the things you've already set up. So you can easily get your data up to any of these cloud providers that are out there. Others that are sort of S3 compatible would also work, but obviously these are the big three that we usually talk about. It also means you can now get your data up to the cloud in an easier way. So straight off the instrument, it can do that transfer for you. So if you're running in the cloud or you're cloud enabled, now Revio can send that data straight to the cloud. And best of all, this is all free to use. So we're going to provide this solution beginning in Q1. You'll be able to sign up. As I said, you need to point it at your cloud provider. So if you've got data storage and analysis or compute style offerings there, you get to carry those forward. There's no middleman, there's no markup from us. We're really just trying to enable access to Revio data and all HiFi data as easily as possible. And to that point, it becomes completely compatible with all our PacBio compatible partners. So folks like DNA Nexus, DNA Stack, Genix, Golden Helix, all of these folks that allow you to do great analysis in the cloud with the workflows that Mike and others will talk about. You can now easily connect these two pieces together and form a streamlined end-to-end workflow. So again, really excited to see us move to the cloud and bring more options for people to get going, making it as easy as possible. And then I'm going to wrap up here in this first little section talking about Revio and the new Spark chemistry, as we call it, which I believe is our biggest innovation since we launched HiFi many years ago. And to see how far we've come in the 2 years since we launched Revio at ASHG, it's really exciting to be here and to update you guys on this. You know, the first thing that this Spark chemistry really set out to do and has delivered on is to just radically reduce the amount of DNA that's needed for HiFi sequencing. And so we're talking about a four-fold reduction here relative to what we launched with on Revio, and it gets you down to about 500 nanograms of DNA to do a true long-read native DNA whole genome sequencing run on Revio. I think talking about 4x and getting it down to 500 nanograms is amazingly impressive, and kudos to our R&D team for making this happen. But if you go back even just a few years before that, and certainly when many of us joined the company in 2021, 2022, we were talking about 15 micrograms of DNA to do a 30x genome. And so that's a 30-fold reduction over the past several years of how much DNA is required. And so if you think about things like enabling saliva extraction to get great high-quality material, lowering the DNA input, all these things start to come together to make it as easy as possible for you guys to take this data and do amazing things with it, which is what you're going to hear about from my colleagues here. The other thing this chemistry did was allow us to boost the output of the system. So now Revio will be able to generate 120 gigabases off an individual smart cell. So that's 480 gigabases in a run. It's about a 33% increase relative to what it was before, and allows you to do two 20x genomes. And Mait is going to talk about some of the great work that they're doing at Tartu at around that coverage. So you can get a feel for what 20x genomes is going to let you do in your labs. And then finally, there's a growing excitement, and I mentioned some of this in the saliva extraction piece, around what we can do with methylation. So not only are we lowering the input, increasing the output, we're also adding and improving the epigenetic performance of the system. So we've improved the performance of 5-methylcytosine calling. It's about a 10% increase in accuracy. And we've also added 6MA for the FiberSeq assay. Folks who are unfamiliar with the FiberSeq assay, please come talk to us afterwards. I'm not going to jump into it now given the time. But I will just say that all of this runs for free on the instrument. Right at the end of the run, you'll get this information out into your BAM file so you can take advantage of it and gain those actionable insights that we've been mentioning. Spark chemistry has also allowed us to radically improve the Revio system itself. And so because of the improved performance, because of all the work that the engineering team's been doing, we've been able to pull a lot of the compute out of the system through optimization. And with that, we can actually pass that savings on to all of you. So now Revio has a new list price of $599K, so about $200K less than when we launched it, if you can believe it. One of the few things in this world that seems to get cheaper. And then about $500 for a HiFi genome on this run. So really excited about where this is going. This is all on the higher throughput side, and as I said, it's great to update you guys on where we've been for the last 2 years with Revio. But hopefully many of you have seen that we also made another announcement here at ASHG, and that is the launch of our first ever benchtop HiFi sequencer, which we're calling Vega. I really hope many of you have come up to the booth and had a chance to look at this thing. It is a wonderful instrument. I'm very excited about the work the team's done. It truly is a benchtop. And if you think about taking all the innovation that we got into Revio and shrinking it down, that's what we've really been working on. So Vega is really the culmination of a lot of innovation, a lot of miniaturization, and a lot of excitement from all of you around how can we get access to HiFi data in a new way. And so with Vega, you'll be able to get 60 gigabases of HiFi sequencing out in 24 hours. And just like Revio, it includes all the great performance features like deep consensus base calling, 5-methylcytosine calling, and demultiplexing to make it as easy as possible. Speaking of making it easier, you know, again, if you go back a few years, it took about 15 different pieces of consumables and reagents to get going with HiFi. We're now down to just 2. So what you can see here is that there's just 2 consumables to load Vega. It really is simple as pipetting your library in, loading these 2 consumables onto the instrument and hitting go, and then you can stream to the cloud to do your analysis. So really excited to bring all this innovation. As I said, I encourage you guys to come up and go hands-on with it. It's a monumental day for us at the company to be able to offer something like this. Lots of innovation and lots of excitement about the data quality. So I'm just going to do 3 quick slides here and wrap up on the data that comes off Vega. So firstly, lots of growing excitement, as you can imagine, around looking at full-length isoforms and using our Kinnex kits to maximize the real estate on PacBio HiFi sequencing. And so the first experiment we did was really to take Vega and to take Revio, run the same Kinnex library, and look at the levels of isoforms in there and do a correlation. And what you can hopefully see on these screens is that that correlation is almost perfect. We're at an R squared of 0.996, and effectively there is no difference between this data. So irrespective of if you're looking at Revio or Vega or you've used some of our data before, it doesn't matter. It's all the same great HiFi. So all those features that you're going to hear about, you can get access to on this new benchtop sequencer. The same is true for our pure target assay. So like I said, if you're interested in repeat expansions and looking at targeted regions in an unamplified way, you can use this assay to really go and interrogate those parts of the genome. Works fantastically on this, and lots of excitement around really looking at those expansions. And Mike and his team have presented many great talks at this conference around all the things that they've done to enable the analysis on this. And then finally, obviously, as we think about 60 gigs of output, it's perfect to do just a human genome in 24 hours. So if you have a sample you want to run through quickly, an extra sample that came in, you can do that on Vega now. As we said, great performance coming out, all the things you'd expect from HiFi, and retaining that methylation information. What I think most people have been surprised by, though, is the fact that we've been able to get this system out at $169,000. So a monumental achievement when you go back just a few years in where PacBio was for all these performance features. So really excited about this product. As I said, please come talk to us about it up in the booth. And with that, I'm just going to wrap up and hand over to Mike so he can talk to you about all the great ways you can take this data and extract the rich information from it to get access to the most complete human genome out there. So with that, I'll welcome Mike Eberle to the stage. Thanks, Mike.
Thanks. So I'm going to talk about everything after the data comes off the machine, and I'm going to concentrate on a couple specific aspects, but I always like to start out by highlighting the whole genome sequencing pipeline that we've developed. And I want to highlight just a few things about this. I'm going to actually talk more about how we benchmark and work that we're doing in that area next, but I do want to highlight a few kind of philosophies about what we're doing. So this is our list of kind of our entire workflow right now for whole genome sequencing and human sequencing. And a couple of the features is that we're actually building all these tools ourselves other than Deep Variant right now, and we're working kind of closely with them on how to improve that. But what we want to do is actually not just have a caller that works it's like a small variant caller, but we throw it at long reads. We want to enable it to work best with the features of our data. The other aspect that I think you'll see in here is that we've kind of highlighted the clinical importance. And so we're really trying to get into the better calling in the dark regions of the genome, and also getting these kind of clinical variants that we need to do, like SMN1 or the tandem repeats. And some of these actually will later on be talked about, I think, by a couple of my follow-up speakers. But I want to actually talk a lot about what we're doing now to actually better benchmark and how we're working to actually improve our variant caller and kind of the process we do. And so this is what we call the Platinum Pedigree. It's a collaboration that we have done with groups at University of Washington, Eben Eichler and his lab, and groups at University of Utah, Aaron Quinlan and Lynn Jordy and their labs. And what we've done is sequence this. Right now what I'm showing here is a 3-generation pedigree. Actually it's actually a 4th generation. I'm not showing the 4th where we got 10 children at that level. And for our group, what we're trying to do is we're trying to build up a better benchmark data and see how we can actually use large pedigrees to actually develop new methods and assess the methods that we have. And what you'll notice also is that we have a variety of sequence data on this. So we're trying to also throw out every type of sequence data, every technology, to try to build up a comprehensive truth set that I'll actually describe I'll kind of go through in the next areas there. So if people are familiar with what was originally called the Platinum genomes about 8 years ago, you might be familiar with kind of the way this works. So what we're doing is we are first building up a chromosomal inheritance map. So based on that, we think that we can look at almost any position in the genome And we can look at the children in the third generation, and we can say which haplotypes they inherited from each parent. And so with that, once we have that, we can actually take any type of genotype call that's made across the family. So we're concentrating on the second and third generations. We can take any set of genotypes and we can say, are they consistent with this inheritance that we already know about? So we've got the biological inheritance. And if they are, we consider that variant to be accurately called across. So it needs to be correctly genotyped in 10 individuals for this. We can do this with the different technologies like I showed. We have calls for our ONT, our PacBio calls with HiFi, and Illumina calls with Dragon. And from that, we identified on the small variant side, 4.9 to 5.6 million variants, the SNPs and indels that we consider true. And so what we want to do is this final call set, we want it to benefit from the strengths of all the technologies. And so then what we do is we merge these together. And we don't require multiple callers to have the same variant as truth. We just require any one of them to actually be correct across the genotype or across the full family. So we ended up, 4.9 to 5.6 million total SNPs and indels. Then we merge them together. The only thing we do to take things from that is actually if there's conflicts. So if there's genotype conflicts, the primary conflict is something like indels where maybe alleles might be called differently due to anomalies in how the alignment's done or factors like that. So in the end, we ended up with about 6 million small variants, 4.9 million SNPs, and almost 1 million indels that we kind of consider our true set. And the goal here isn't just a SNP and indel call set. We're also looking at structural variants and tandem repeats. We've repeated the same procedure for both of them. For structural variants, we're just using the long read data, both read-based and assembly-based calling. For the tandem repeats, we're using we're using target. And that delivers the entire sequence context from that. And so you'll notice now what we have is the numbers have changed a little bit, and this is because when we kind of combine this all together, there's many times where an indel will overlap with a tandem repeat call. So we're actually keeping those in the tandem repeat bucket, so some of the numbers went down. One final thing I'd like to say about this, just in case people are wondering about the numbers, this is truth based on a family. And so this isn't the number of variants you see in one individual. These are number of variants you would see in two individuals. So they are a little higher than you would normally expect. So as we're building this, there actually are other benchmarks out there. And I just want to kind of, pay homage a little bit to the other, kind of some of the top ones out there. We are doing this on this one pedigree, so we would call it NA12878, kind of our truth benchmark. Genome Metabotle is currently on HE002, and the T2T is on HE002. The important thing is, as all of these actually have different strengths and weaknesses. So Genome Metabotle wants to be very, very precise. We want to be very, very comprehensive. And T2T in the end is also kind of getting towards both of those is the hope for that, but I feel like using the pedigree actually adds a lot of value. And I think that the big take home from this is that you'll actually see, as you look at different truth sets or benchmarks, you see a wide variety of calls. So the indels and the SNPs can both vary by about 400,000, between the smallest number and the largest number on that. One of the things, in addition to just being able to understand like accuracy and levels like that, one of the big points of this data set is actually for us even to just improve the variant calling. So whatever level we're at, we can actually use this to try to better train. And so we've actually done some stuff with the Google team with Deep Variant, which is our primary small variant caller, where they had originally trained on the Genome Metabotle. I've actually shown kind of an overlap in NA12878 between the total benchmark or truth calls. And you see that most of the variants are shared between them, but we actually have we have almost 800,000, 760,000 variants that are unique to this method of identifying it. There's also ones that are unique to the Genome Metabotle. And the question is, though, as we build this truth data set up, there are conflicts, there's things that are a little bit different. Ultimately, does it actually help the total performance. And so the group at Google actually retrained with this and used it as a comparison with their previous one. And so they found that they were able to reduce their errors by 33%. So this is like a big jump in what we're doing. And I think I would just even say, I don't think that's I think this is the first start. So it's actually identifying that there are, as we build up better and better truth data, the models that run and call variants can actually be improved significantly on that, just even in a deep variant AI type method. Another thing that I'm very excited about doing with this that we're highlighting here is when you actually develop new methods, one of the problems is like what is the ground truth? And so for things like SNPs, indels, and SVs, even though that can be complicated, you can still kind of get that. When we're actually going out beyond that, many times you just have to find a sample that has some orthogonal validation, like when we're doing tandem repeats, we need to find that. But as a developer, having this pedigree and knowing the inheritance actually gives us a first step, even just at the very early stages of looking at this. And so what I'm showing here is work that we're doing, a caller that's in progress to call KIV2, the KIV2 repeat. So if people are, I wrote a little bit about that there. If you're not familiar with it, it's about, the repeat motif is about 5,000 base pairs long. It's repeated 20 plus times. And so it's 100 KB long in total. And so we developed what I would call a kind of a guided assembly for that, because there actually are kind of informative sites within this that we can key off of. And what I'm showing here is we ran this across this pedigree, and what you can do is look at how the links that we're calling are inheriting within this family. And what you see is I've color-coded these by the haplotype. So the mother, 77 would be the father, has the yellow and the blue. And the mother has the green and the kind of purplish. What you see is all the ones that inherited this purple haplotype from one of the grandparents eventually, they all have this really long allele. We also see the ones that carry the short allele, the blue haplotype, that is all consistent. And so this is just the very first level, gives us a really good way to say what we're doing looks good. Then we can actually kick off to the next step of like, maybe we want to look at a lot of trios, maybe we want orthogonal data. But this is a really good way just even to kind of QC the general premise behind the methods that we're doing. And actually, one last thing I will say. All of this data that I'm talking about, other than 3 samples that aren't consented, is all publicly available. And so we have a paper on bioRxiv if people want to kind of download that and look at it. The last thing, I'm actually going to go through this very, very quick because I'm not going to talk about this. Some of the new variant callers that we're developing, there's 2 that are being presented today, and I think actually one of them might be talked about by Mait after me. But Chris Saunders is going to talk about a new variant caller for structural variants called Sawfish. We really like the performance of this, and I encourage you to go and talk to him and ask any questions about that. Matt Holt is going to talk about some of the work that we're now doing on PGX with a caller called Starface. And I think Mait is actually going to show some of the stuff from that too. We actually are looking at all the class A genes with that. And feel free to run by him and ask him questions at the poster. And then the last thing is the callers that I walk through. Many of these are, as we develop these, we collaborate deeply with people to, get the right data and validate it. We're also publishing on these. So a couple ones I didn't talk about in that big thing and that have been presented at talks here are Paraphase to look at segmental duplications. There's a publication on that out and a new one coming out in bioRxiv. It's in bioRxiv, hopefully coming out peer-reviewed soon. Our tandem repeat genotyper target, also with some recent work with people at the Broad, and HiFi has been published there. So I encourage people to either come up to me, talk to people, or check out our articles on some of our previous stuff. I didn't include, I'm sure I missed a lot of people, but I just wanted to highlight for the Platinum Pedigree that the work that I'm kind of presenting here is collaborations with groups at University of Washington, groups at University of Utah, and we really appreciate working with those, and we appreciate working with a lot of the other more clinical people that I didn't even get into some of their work. So I will now hand it over to Carol, and she will go maybe talk about real work on this stuff.
Hello, thanks for being here, and thanks to PacBio for the opportunity to tell you about our group's work. And just by way of disclosure, they covered my travel funds for ASHG. So we're in Kansas City. Just a bit off to the east here is where the Kansas City Chiefs play, where Taylor Swift may or may not be hanging out. And she has been spotted at dinner right up on this hill here. So it's all happening here. And there's a lot of genomics happening on the 3rd floor of this cool new building that we're in. And it's a unique setup where we are together with research and clinical in the same space, which really enables great translation flow of new technology into the clinical space. So most of my focus is, well, actually most of our focus in general is on rare disease because that's the majority of our volume. We do also some oncology, but what I'll focus on today is rare disease. And so we all know that we fall short of finding a diagnosis for well over 50% of the patients, despite our best efforts with exome sequencing. Short-read genome sequencing adds an incremental boost, but not much in terms of diagnostic yield. And so when we look at what NGS has done to really transform genetic medicine in the way of new gene discovery, that's been a huge thing in improving our diagnostic rates and decreasing the time to diagnosis. So we do really well with single nucleotide variants from short reads and also copy number calling. When we get into expansion disorders, methylation disorders, structural variation, we fall short with short reads. And there's also mitochondrial genomics to consider. So there's just a lot of complexity to trying to make a diagnosis for these patients. And if you're like me and you run a clinical lab, you have a whole lot of different workflows that you need to complete all the different testing that people need. And each step of this pyramid takes time. And if you're like us and you perform insurance pre-authorization for outpatients, then that can add up to a month in between all these steps. And that's if the physician is thinking of the right test to order for the patient based on their phenotype. So in a case like a Down syndrome where it's a specific phenotype, it's an easy call to do karyotype. That's one thing, but there's a lot of workflows that are more complicated than that. And one example is congenital hypotonia, where you might have up to 7 different tests that you need to get to the right, or more, to get to the right diagnosis. So it's a pretty common indication that we see. It affects about 0.8 in 1,000 births. There's multiple genetic mechanisms that complicate this. So, you can have targeted genetic testing looking for spinal muscular atrophy, myotonic dystrophy, and Prader-Willi syndrome. And none of those things are very well detected by short-read sequencing, which is what we tend to counsel our ordering providers to start with as a first-tier test for many patients who have just kind of an absence of specific features to guide them to any specific test. But if you do that, you're going to miss most of this arm of this testing algorithm. And in order to have treatment available, so for some of these things have treatments like SMA, spinal muscular atrophy, you need to have a diagnosis. So that's very important. So we have consolidated this pyramid of testing down a little bit. We've largely gotten rid of microarray in favor of doing CNV calls from short-read exome or genome sequencing, but still there's a lot of steps here. And so we really wanted to look at long-read sequencing as a solution to further consolidate our testing. So we can't control the modality of testing that is sent to us. So in a lot of cases, we get singleton analyses, which is really going to limit our ability to detect de novo variants. But it also is if you don't know the phase, then you're going to miss a lot of diagnoses for autosomal recessive conditions, which comprise about 20% of cases. We also struggle with coverage in short-read genome sequencing, even if it's PCR-free genome sequencing. So long reads are better at all of these things, including looking at repetitive regions, pseudogenes, expansion disorders, structural variation, like inversions and translocations. So again, all this just can be time-consuming and limited to what you know to look at if you're not just doing it all in one test, like a HiFi. So we wanted to look at what does this look like if we're using HiFi for these types of disorders. So just to give some background, we have a unique research program called Genomic Answers for Kids. It's run by Tomi Pastinen, and Isabel Tiefelt, Emily Farrow, and Anna Cohen do a lot of work in this space with him. So their mission is one of their missions is to make a pediatric data repository to facilitate the search for answers and novel treatments for pediatric genetic conditions. So they began a collaboration with PacBio back in 2020, where we have a big repository of patients with rare disease who need a diagnosis, and if they have already had short-read genome or exome sequencing, they get reflex to HiFi sequencing. So we've now done over 1,500 HiFi genomes of families with rare disease. A lot of this data was published a couple years ago and made publicly available, because as you know, when you have a new data set and you've got a whole new set of variants that you've never seen and you've got no data set to filter off of, that becomes a huge challenge for analysis. So we made our own. And so with this research experience, they demonstrated equivalent or superior SNV and CNV variant detection. Here's short-read genome sequencing sensitivity, and HiFi sensitivity is a bit higher. So for each proband, you see about 150 rare variants that you don't see from other technologies, and we're also able to see methylation and repeat expansions. So our analytics strategy employs several tools. So things from PacBio, we use deep variant for SNV calling. We use WhatsApp for phasing, and PBSV for structural variant detection. Tandem repeat genotypes are annotated with targets. We use the kinetic embedded algorithm for methylation detection, and our informatics folks have trained this methylation data on just a load of samples that they've run and compared what does normal look like statistically versus someone with a methylation disorder. And so they put out these nice reports for us. And then we use Paraphase to evaluate 11 pediatric relevant loci that are encumbered by pseudogenes. And all this is in the cloud, Azure cloud. So when we were ready to move this to clinical validation, we ran samples on the Revio. We included cultured amniocytes, cells, and blood, and we sequenced to a target of 90 gigs. And we included, so we gave them blind samples with known genotypes that represent, disorders caused by repeat expansion, CNV, SNV, complex loci, and methylation disorders. So our validation statistics pretty much held up what they found on the research side. We have higher sensitivity and specificity than short-read sequencing. Our expansion testing had 100% concordance between the HiFi target and orthogonal methods used to detect repeat expansions. We had all of our complex loci that were interrogated by Paraphase were detected. And methylation analysis was able to detect 4 of 5 known methylation signatures. We fell short at hypermethylation at ICI1, which represents a rare cause of Beckwith-Wiedemann syndrome. We began with inpatients with severe indications such as multiple congenital anomalies, hypotonia, overgrowth, brain malformations, seizures in infants, and regression. And as we got going, we kind of expanded these indications to include other indications that are listed below in red. And we just recently began also doing some outpatients on a case-by-case basis. And so we recognize not everybody needs every tool in the toolkit. So depending on the patient's symptoms, we kind of cater what we need to analyze for each patient. So the only indication where we do all of these tools is hypotonia versus hearing loss or cardio concerns. We would look for CMVs, SMVs, and use paraphase to look at key genes that we might be missing with short read. So I'll just walk you through a couple cases that required HiFi sequencing. So this first one is a 2-day-old male who was he was in the NICU for respiratory distress and pulmonary hypoplasia, had some dysmorphic features, including bilateral club feet. And this is what the report from our informatics team looks like. So you can see this is like the average number of repeats a normal person has. You can see his are in the expanded range, consistent with what you would have for congenital myotonic dystrophy. So this is DMPK repeats, and the mom is also affected. So in IGV, you can see not only the repeat, but you can see the patient has hypermethylation of DMPK. And what I want to point out is this patient was not referred for hypotonia, which would be one of the main drivers for ordering specific DMPK testing or a hypotonia panel. So for a case like this, in the absence of some family history or something else to point to DMPK, this diagnostic odyssey for this family would have probably gone on for quite some time if you hadn't done an all-in-one test like HiFi. This second one is a teenage girl who had pseudo-hypoparathyroidism, and we're able to show very clearly she has aberrant GNAS methylation. So this is kind of the curve of what you expect for methylation in our large cohort that we run, and she is off the charts here, and you can see in IGV she has hypermethylation. And then the third one is this is a 2-week-old male who had brain anomalies and microcephaly and was found to have an unbalanced translocation, resulting in a 6q deletion and an 18p duplication. And all in one test with the parents, you're able to accurately map these breakpoints and show that the father carries an unbalanced translocation. And the feedback from the clinical folks is that it is just so nice to have this efficient testing strategy where they just have it all in one go and don't have to chase down parents for follow-up karyotype or something like this. So with HiFi genome sequencing, we are able to consolidate our testing, resulting in a higher diagnostic rate, a quicker time to diagnosis, and ending the testing odyssey. And it's just a really nice, efficient algorithm that saves both clinician and laboratory time. And while it might be nice if you had a crystal ball to know which patients really need it, you don't. And what I see happening already is patients have negative short read sequencing, and then the clinicians are already asking, can we have also HiFi afterwards? So nobody has done the numbers to crunch what that looks like, financially. But I would also leave you with the thought that our research cohort, our research team identified a patient who had a cryptic SMN1 variant that was undetected, like he went undiagnosed for 2 years, and they diagnosed him right around the age of 2 and were able to start treatment for him finally, because you can't have SMA treatment without a diagnosis, but he passed away shortly after. So I don't know how many cases like that it takes to make a compelling argument for this strategy. But our future directions are to expand this to more patients, look at more methylation loci. For now, we're restricting it to only 11. And we're beginning to do oncology samples or validate. We're working on validation for that. So we're looking at leukemic blood, tumor normal pairing, and solid tumor. So I really want to thank our whole team, especially Emily Barrow and Isabel Tiefelt, who did the legwork on this, and Tomi Pastanen, who directed it on the research side, and our whole informatics and lab team. So thank you very much. Sorry.
Hello, everyone. Good to be here, and great that you are here. I'm Metspalu. I'm from Tartu, Estonia, and I'm going to be talking about a little bit from a different point of view than the last speaker. I'm talking from the university setting, and actually I'm mostly going to talk about what Estonia as a state is doing in terms of implementing personalized medicine. And by personalized medicine, I mean mostly personalized prevention using polygenic risk scores. Personalized medicine is, of course, much wider thing, as we just heard, and also cancer. So there's disclosures. There will be stuff that has been, let's say, sponsored by both PacBio and Oxford Nanopore. So the background for Estonia in terms of personalized medicine is that we run the Estonian Biobank, which today has over 200,000 participants. Compared to the population size of Estonia of 1.3 million, it makes 20% of the adult population is in the biobank. We have genotype data for all of the participants, some other data layers. And of course, important is that we have retrospective health data from different health registries from Estonia. And we update these data on a regular basis. We get diagnosis, we get, for example, health insurance bills, which means that we see if a person actually bought the medicine that was prescribed. We have clinical measurements and so on. And it is accessible, so anyone who wants to do research on it, just find us. And of course, lots of science have been conducted on these data. Another important thing we promised out when we started the biobank is that we give results back to people. And this has been a very long journey. Finally, earlier this year, we launched the Biobank Participant Portal, where we give back polygenic scores for type 2 diabetes and cardiovascular disease, pharmacogenetics report for 37 active ingredients, and a genetic ancestry report. The cool thing, I think, about the risk scores that we gave is that we just don't give a static risk score, but genetic risk score, but we combine it with lifestyle risk and show it in a form of risk trajectory, which you can play with if you alter your lifestyle, numbers. In this case, it's type 2 diabetes, and you can change your, let's say, weight. Let's say make a target to lose, let's say, 10 kilos, and you can see what that does to your combined risk for type 2 diabetes. This particular example here is a super healthy person, probably a non-existent one. One of the sort of things that caught a lot of attention in the society was, of course, the genetic ancestry report. As I mentioned, I didn't mention it, so already 100,000 people have logged on and checked their results. This is half of the biobank and 10% of the adult population of a country. At least from social media, it appeared that the country stopped for a day and everybody was reporting their Neanderthal percentage. And we also give a look into within Estonia genetic ancestry, and people were quite excited to look into this stuff. But this is, of course, not personalized medicine. This is this biobank giving back their results. Although we use legally, what we use is in-house medical device, but that's beside the point. So what's going on the country level is that you can see there's a project that was run from 2016 to 23, where which was run by actually by the Ministry of Social Affairs. Tartu University was one of the main partners, and the goal there was to actually think through how genetic data can be and will be used in terms of personalized prevention in medical setting. And we came up with a IT infrastructure coupled with the legal framework such that there is a centralized, do I have a pointer? Yeah, okay. So there's a centralized data repository where different data providers, data owners will deposit their data, be it the biobank, us, or medical labs. And then there's a secure computing environment where medical devices will come and meet the data, calculate, get results, and then these results only go out to be used in the medical service. So this is, I think, the key aspect here is that the data doesn't go to service providers, because there will be several or many service providers, and we want the data, or actually everybody wants the data to be safe. So based on different clinical studies that have been conducted in Estonia, the Estonian Health Insurance Fund, which is the financer of publicly funded medical service in Estonia, has decided that it makes sense to finance breast cancer screening program that uses genetic data. Of course, we have breast cancer screening program for years, but now it's going to be what's going to start early next year is that genetic information is polygenic score for breast cancer is included in the screening program by stratifying women so that the ones with high genetic risk will just go to the screening program earlier. So this is actually happening, which is cool. But there are many hurdles on the way. One of the sort of unifying hurdles is that these different stakeholders, different parties that are involved in implementing personalized medicine, they are quite often still working in isolated silos, and we've created a new structure that's Center for Personalized Medicine that unites those, and we attracted funding for this, and one of the targets or one of the goals of the center is to actually create a exhaustive reference panel for imputation for the Estonian population. Unlike the Emirati genome project that will sequence everybody in Arab Emirates by the end of next year, as I understood, we don't have that capacity, let's say. And in the sort of in the coming years, we see that we aim at widening the genotype level data or low-pass sequencing data for the general population, and for that, of course, then you would need to impute it, and for that you need a very good reference panel so that you can actually offer service for every, or at least vast majority of the citizens. It's not a sort of research question where you can filter out the ones that you can't impute, but you need to be able to offer service for more or less everyone. So initially, we're planning to do combination of long and short read sequencing, but as the evolution of price of sequencing was supportive, we gradually started to lean to the idea of sequencing all of the 10,000 genomes with long reads, for obvious reasons that are listed here and have been talked about. Then, of course, there was the question: should we go with Oxford Nanopore or PacBio? And up till now, you probably thought that you ended up in the wrong room because he's not talking about PacBio. So what we did was that we wanted to compare the two technologies using samples from the biobank, so that we actually used the samples we're going to use the technology on. Because some of the samples have been collected in 2010, 2018, so some time ago, and we wanted to be sure how they work. And these experiments were then kindly supported by both technologies, both companies. So what we did, we took 16 samples from the biobank and performed sequencing. And we aimed at the 30x coverage at this test, and which was achieved, of course. You can see that the median read length is shorter for nanopore because the DNA was actually fragmented for that experiment for the reason of trade-off between yield and read length. And also the percentage of bases reaching Q30 is higher in PacBio. Then what we did is that we compared the results against the Illumina sequencing we had available for the same samples. It's not ideal. We're not looking at up to 10% of the genome that is not accessible by Illumina, but this is what we could do. And you can see that, and we have PacBio 30x, PacBio 20x was in silico downgraded, and ONT here. For SNVs, you can see that basically there's a slight reduction of recovery of Illumina identified sites in ONT, but that's a small reduction. It has some clinical meaning, but not too much. But when you look at indels, then the difference is quite huge. We are recovering much less indels with nanopore than with the PacBio. And this also has some clinical relevance. This is summary of the two comparisons in terms of SNPs. Both technologies are actually doing very well. In terms of indels, you can see that the recall rate of ONT is significantly smaller. So PacBio was the answer. And as I said, the main reason why we set up sequencing 10,000 genome, the sort of what we wrote in the project and got funded for was to create the exhaustive imputation panel. But of course, especially doing long-read genomes, there are many other things we can do and are doing. Among those, so of course, assess the complex configurations of important pharmacogenes, overall structural variation, and of course, methylation data that has been mentioned. So as the first goal, the main goal is to get the imputation panel. It's important how we choose the samples. So we rely on the biobank because we know quite well how the biobank represents the Estonian population. So the strategy was to k-means clustering to arrive at 20 clusters first, and then within each cluster to do hierarchical clustering, so that we arrive at called branches, and then within each branch we can choose one genome, and the branches can have one sample or several. And if there are several samples, then we can use downstream criteria to choose the genome or sample to be sequenced, be it then hints of monogenic disease-causing variants from previous data or structural variation in pharmacogenes. And this strategy for choosing the genome was done and devised by Redikmägi and Martkals, and Martkals is sitting over there. So this is just an example of the complexity you get in probably the most famous pharmacogenetics gene, SUP2D6 and D7, where you get duplications and most importantly get gene fusion, which is super hard to detect. From the early results, we can already see how we are getting much more structural variation out of the data compared to Illumina data, which is of course expectable. Current situation is that we are running around 100 samples per week, or sequencing 100 samples per week. As I said, target is 20x, and currently we're still doing manual prep, but we'll upgrade to automation there too. And up to date, we have sequenced already 1,000 genomes. As you can see, this is very much an, sort of not an ongoing, but we are in at the start actually of the thing. And to sum it up, we are also the newest members of the HiFi Solves Consortium, which is a very great initiative to understand how a single, let's say, genetic test, which is long-read sequencing of the genome, can replace many individual genetic tests. So, and with that, I'd like to thank you.